All right, let's get started on this next lecture on the neuroscience of various memory systems. In this lecture, I'm going to introduce a variety of memory systems that I've been kind of gradually introducing all along during the series, and then dive a little bit into what the memory structure system itself is and the underlying structures that support it. A couple of lectures ago, I basically gave an introduction to some of the important structures. Now we're going to kind of start putting this all together in the next couple of lectures. This lecture will look at the systems, and in particular, we're focusing mostly on amnesia patients and the evidence they provide about memory systems, as well as some other selective populations like people with highly specific or highly superior autobiographical memory. In the next lecture, we'll get into fMRI studies of memory encoding and memory retrieval, and then following that, we'll get into some of the underlying cellular bases of memory and talk about consolidation theories of memory. So here's how this will work today. In this lecture, we're going to get into each of these memory systems. I'll first introduce something about those and then talk about the underlying neurobiology. For procedural and memory and priming, I will finish that with a discussion of implicit memory circuits. So let's dive into episodic memory. So these are often referred to as what we call hippocampally dependent memories. That is, they require the hippocampus both for their creation and their retrieval. And a couple lectures from now, we'll get into the specific models of memory. In particular, I'm a fan of multiple trace theory from Morris Moscovich, which delineates semantic memories from episodic memories in the way that I'll be discussing today. These memories are also known as context-dependent memory. The role of the hippocampus really appears to be tied to context, that is, times and places. And there's a great deal of neuroscience in this area looking at how the hippocampus is involved in place memory. There's a really brilliant Nobel Prize winning study on London cab drivers where they demonstrated that there is an increase in specific hippocampal areas and related temporal lobe structures as London cab drivers learn their way around the city, so very clearly tied to place. We also see animal studies that demonstrate this particular phenomenon, as well as if we look at topographic amnesia patients. And finally, Alzheimer's disease patients start to lose some of these structures and oftentimes have that kind of sundowning topographical amnesia, which we talked about in a previous lecture. So let's get into some specifics of episodic memory. In particular, remember this is memory for life's episodes, which you did yesterday, which you had for breakfast. Based on patient HM, we know that the hippocampus is critical for the formation of new memories. So patient HM, of course, had most of both his left and right hippocampi removed, along with some of his medial temporal lobes. He was no longer able to form new episodic memories. That is, he wasn't able to form any new memories of his experiences. He did learn. He had intact procedural learning, semantic priming, and even some intact semantic memory learning, but pretty degraded. So mostly his episodic memories were involved. We have another patient named RB, and RB had brain ischemia following a cardiac incident. And interestingly, while I was researching this particular lecture, I found some animal research showing that this is a common side effect of heart attacks in individuals is some loss of CA1 pyramidal cells due to ischemia. And so there's some interesting questions about how heart attacks might affect risk for memory loss. But in patient RB, what we can see is his hippocampus on the right, and very specific selective loss of the CA1 subfield of pyramidal cells of the hippocampus in patient RB, and he also was no longer able to form new memories. His retrograde amnesia was of a less extent than patient HM. Both had some loss of prior memories. But RB has an inability to form new memories. And as I spoke in a previous lecture, the CA1 subfields of the hippocampus are particularly susceptible to anoxia or lack of oxygen. And so that loss of a heartbeat during a heart attack or a cardiac incident or cardiac surgery is not surprisingly having this deleterious effect on the CA1 subfield. So this is the particular part of the hippocampus that seems to be really critical for forming new memories. This is CA1 subfield of the hippocampus. So we also know that damage to the lateral anterior temporal lobe without hippocampal damage can result in isolated retrograde amnesia, which is relatively rare, but we do see cases in herpes simplex encephalitis where people have pretty significant retrograde amnesia. The case of Clive Waring, he had both retrograde and anterograde amnesia, extensive damage due to herpes simplex encephalitis. And so something to understand about that is that anterior temporal lobe cortex, the lateral anterior temporal lobe cortex, appears to be particularly important for previously learned memories. Whether they're stored there or this area is responsible for their retrieval is a bit of an open question, but it's certainly an important area for those memories. Finally, patients with major depression and depressive symptoms have been shown to have reduced hippocampal volume and reduced memory. I would refer you to some of my lectures from my Drugs and Human Behavior course. In particular, there is a lecture on non-SSRI treatments for depression. In particular, there's some really terrific research involving mice. Oh, it's a mouse study, sorry, but it's involving ketamine treatments for major depression and major depressive disorder. So the major risk factor for depression is stress. And so in this particular study, they used cortisol injections as a stress inducer, because that's what stress does, is it causes the release of cortisol. They saw reductions in CA1 subfields of the hippocampus, reduced hippocampal volume, as well as some other dendritic branching loss in some other areas. In particular, in the hippocampus, it was a loss of neurogenesis. Treatment with ketamine reversed that damage relatively quickly and actually improved both memory and behavior in the mice themselves. Really interesting study from Science, and so take a look at those studies. Really interesting stuff. What we see is that reduction in hippocampal volume is directly associated or correlated with reduced memory in major depression patients. So important thing to keep in mind about depression is it's not just a mood disorder. It does have effects, pretty significant effects on cognition. So that gets us to semantic memory. Semantic memory is our world knowledge or stored facts about the world. And our semantic learning, we know, starts as episodic memory because it is linked to particular contexts. In a study that I conducted at George Washington University when I was a postdoctoral fellow under Elliot Hirschman and Mike Berrigan, we found that as we had people learning new semantic facts, these were obscure facts like Donald Duck's middle name is Fauntleroy, things that most American students wouldn't know. And what we found is that if under degraded memory conditions, that is, they were given midazolam, which reduced their memory, they were better able to retrieve those facts if they relied on their episodic memory. That is, they were told specifically to retrieve facts they'd learned earlier. And even though they had very little episodic memory for that period, they were better able to do so under those instructions than just simply being asked to answer any questions they might know the answer to. And so we see that episodic memory supports semantic learning early on. And we'll take a look at this a couple lectures from now when we start looking at consolidation and multiple trace theories of memory. So eventually, these memories will become hippocampally independent. That is, they are no longer dependent on context. And so your semantic memory is no longer tied to when or where you might have learned a fact. And so we're independent of the hippocampus and some of the medial temporal lobe structures, not all of them. In particular, what we see is the pararhinal cortex, which is part of that medial temporal lobe complex, is particularly important for semantic memory. So early on in learning, you can see these reciprocal connections between pararhinal cortex and anterorhinal cortex and the hippocampus, as well as the parahippocampal cortex. As we start developing a more advanced understanding of memory, we believe that the parahippocampal cortex is more involved in those episodic memories, where the pararhinal cortex is more involved in semantic memories. And so early in semantic acquisition, the hippocampus is involved. Eventually, semantic memories are stored and retrieved independent of the hippocampus. And so that pararhinal cortex to frontal parietal, etc., connection is how we get retrieval of semantic information. Interestingly, if you look at Alzheimer's disease patients compared to semantic dementia patients, you can see pretty clear delineation of damage to these structures. So on the left, we have a semantic dementia patient, and on the right, an Alzheimer's disease patient. Regions marked in red are the anterorhinal cortex, and in blue, the pararhinal cortex. And so what you can see is dramatically reduced pararhinal cortex in the semantic dementia patient that is not seen in the Alzheimer's disease patient. And one thing to be clear, these are radiological profiles, so the left is on the right. So these are left anterorhinal cortex and left pararhinal cortex. And of course, the left hemisphere is primarily involved in verbal material. And in fact, visual spatial material appears to be encoded and retrieved more in the right hemisphere, and verbal material more in the left hemisphere. But really dramatic, showing that semantic dementia appears to be pretty tied in with that pararhinal cortex. That gets us to autobiographical memory. And autobiographical memory is a bit of a hybrid memory of our personal life stories. That is, it is both episodic and semantic in nature. That is, we have stored facts about ourselves. I grew up in Colorado Springs and Canyon City, Colorado. I went to Canyon City High School. I went to college at Colorado State University. These are facts about me. You have facts about you. And those facts are not necessarily tied to specific life episodes, but we oftentimes use those facts about ourselves to guide memory retrieval. And so we think about a period of time in our life. Well, I think in that year I was living at this address, so I have these memories that are associated with that experience. And so it's a hybrid of both semantic and episodic memory. One of the things that we understand, and we'll get into this relatively quickly when we start getting into social cognition and emotional memory, is a lot of our sense of self is made up from our narrative identity. That is our life stories. And there's some really interesting work done on our narrative identity, and we can actually use this concept of narrative identity to try to resolve issues from our own lives. So there's a lot of clinical work being done in this area, in particular about trauma. And one of the things that really helps with trauma patients is getting their story to the point where it's integrated, it's part of their life, but it isn't traumatic. And so it's just part of their narrative, it becomes part of their narrative identity as their life stories. And so basically you're sort of writing the next chapter is the way to think about it. So autobiographical memory, as I said, is a hybrid of both episodic and semantic memory. It includes facts as well as specific episodes. What's interesting is there are people who have highly superior autobiographical memory, which is pretty amazing, although I don't know that I'd want to be one of them. There's a terrific video you can find that on YouTube from, I think it's Nova, called Memory Hackers. And they actually show some of these people with this highly superior autobiographical memory. Actually, Mary Lou Henner from the TV show Taxi, some of you may remember her, is one of these people with this kind of highly superior autobiographical memory. They can remember in exacting detail every single moment, every single day of their life. You can ask them June 21st, 1992. They'll tell you what the weather was. They'll tell you what they were doing. They can, if they're a sports person, they can tell you what the sports score from whatever game might have happened that day. It's really, really remarkable. I wouldn't want it because remember, every single memory is exacting in its detail. And so there's some memories and some things I don't want to remember in that kind of detail. So what is autobiographical memory? Well, first of all, there's some evidence that these HSAMs, as we call them, and Jim McGaugh is somebody who has done quite a bit of this work, as well as Kathy McDermott and some others, Roddy Rodiger at Wash University in St. Louis. There seems to be some greater, basically it's about retrieving memories. It's not that their memories are stored better, but they're better able to retrieve them. And there are some connectivity studies that are starting to come out that seem to support that. Kathy McDermott conducted this really interesting meta-analysis of fMRI studies comparing autobiographical recall to sort of laboratory tasks. And so I want to take a look at that data. And so in blue are the lab-based studies. In red are the autobiographical studies. And the sort of brightest green are areas that overlap between the two. And one of the things you can see is the lab-based studies really involve the dorsolateral prefrontal cortex and some other areas like that a great deal more than the autobiographical memory studies. We do get some left temporoparietal junction, probably because a lot of those studies are verbal in nature often, depending on the type of study that they're talking about. One of the things you see from the autobiographical memory is you do see some more occipital lobe, as you can see in the medial view there, because autobiographical memories appear to oftentimes have a more visual component to them. And so we see that also in the right medial view there at the temporoparietal junction. Again, visual spatial aspects to those memories. So we see very different areas being involved in the retrieval of these particular types of memories. And so there do appear to be something more than just episodic memory. There's something more to autobiographical memory. And so there is this emerging view of this parietal memory network that you see with the autobiographical memory. And so we'll take a look in more detail in the next lecture at some fMRI studies of memory and what we've learned about memory. That gets us to procedural memory. This is a form of non-declarative memory for motor skills and tasks, that sort of thing. Piano playing, tennis playing, that sort of thing. In the lab, we use this sequence learning task where people learn to sort of basically tap out sequences with their fingers. They appear to be random, but some of them repeat over time. Participants usually report not being aware of the sequences. That is, they think that they're random. They don't remember learning them, although they demonstrate learning, but this isn't always. It's one of the things to keep in mind. This apparently occurs through that cortical basal ganglia circuit, and again, I refer you back to my motor learning lecture on that particular phenomenon. So what's interesting about this is it's basically goes from the basal ganglia to the cortex, but there are no reciprocal connections from the cortex, and we'll talk about that here in a moment. We do see that Parkinson's disease and Huntington's disease patients show impaired sequence learning in this kind of procedural memory. And so that's something to keep in mind as we start thinking about the structures that are involved. Again, primarily the basal ganglia, particularly for Parkinson's disease. This gets us to priming. Priming occurs when we process a stimulus, or processing of a stimulus, is facilitated by the prior presentation of the same or similar stimulus. And basically, we see this by increased reaction times. People are faster at identifying a particular stimulus, or they are more accurate at identifying what a stimulus might have been. And so this is the basic way in which we do a priming study. We present a prime for 33 milliseconds. We then include a backward pattern mask. And the reason for that is to basically keep participants from using their sensory memory, their iconic memory, from processing that prime by covering up that visual display. And then we get a probe, and that's where we get the participant response. So this is the basic way in which we do various kinds of priming. So this would be a repetition priming experiment. And basically the subject's task would be to indicate whether the probe on the far right was a word or a non-word. So this would be a repetition priming experiment. Apple would be presented with backward pattern mask, and then Apple would be presented again, and the participant would indicate word or non-word, usually by a key press. And then we might have school, then Apple. And then we would have trials where there would be a non-word. What we find is participants, when the word is repeated, are much faster and more accurate at identifying the word, even though they don't. They report not being consciously aware of the fact that the item had been presented. Now, in a semantic priming experiment, we might present a related word. And what we see in a semantic priming experiment is that people are faster at identifying the word apple if the word banana had been presented on a previous trial, because the words are related in terms of their meaning. And so that would be a semantic memory trial. So that gets us to how this works. We have a perceptual priming system that acts within what we call the perceptual representation system. So this would certainly be the repetition priming. We see that amnesic patients show intact perceptual priming on indirect tests of memory. So we talked about that when I talked about amnesia. study by Graf, Squire, and Mandler, sort of the groundbreaking study in that area. We also see that perceptual representation has been doubly dissociated from episodic memory, so there are different systems involved here. Patient MS had a right occipital lobe lesion and did not demonstrate perceptual priming, but patient HM, who of course had no episodic memory, was able to, did demonstrate perceptual learning. So all of this goes under the, both perceptual priming and procedural learning go under the sort of general umbrella of what we call implicit or non-declarative memories. So both perceptual priming and procedural learning involve these subcortical circuits. And so we get sensory and motor information involved that then go to the basal ganglia, the ventral thalamus, and the premotor cortex. What we don't have are reciprocal circuits coming back from the cortex. So they're not reciprocal circuits. And that might explain why these memories are unconscious, because these aren't in two directions, these are unidirectional. And so that lack of reciprocal connection may be the issue. All right, well, that is an introduction to these memory systems and their underlying neural substrates. In the next lecture, we're going to get into neural imaging of memory systems.